Good morning. I just want to make one announcement to you. The announcement is posted to my right. For those of you who, for whatever reason, have to drop this class or need to withdraw from the class, it has to be done by this Friday. If you wait longer to do it, as you were told in the very first lecture by the course administrators, then the campus will charge you a fee, and you'll also have to see a dean. So if you cannot take the class and you're enrolled in it and you want to drop it, do it by this Friday so that you're not charged an extra fee. Today what I want to do is to continue to talk to you about the invasion of the land by plants, the evolution of plants. We mentioned last time and the time before that one of the great strategies that the plants seem to have used was the development of this very unusual life cycle. It's a really difficult life cycle conceptually for you to understand, this alternation of generations with two different plants. And what I'd like to do is to return to the end of the last lecture, to return to the female pine cone, and I want to just spend a few more minutes talking about what's going on the scale of this female pine cone. Recall that we said, if you could imagine, that the pine cone represents a collection of sporophylls, a collection of spore-bearing leaves. And I showed you the fern, and I asked you to try and imagine collapsing all of those leaves down into this cone. So what I'd like to do now is to focus a little more, and for some of you this will be a review because I spoke about this last time, on the scale and ask you what's going on the scale. We're going to take one of these scales, we're going to cut the cone down the middle, and we're going to look at it. So this is going to be the scale of the female cone, and this scale is a megasporophyll. That is, it is a leaf which bears sporangia. So let's draw the scale, and I want to spend a few more minutes talking about it. Here is the scale. And we're going to start from the point where we have a megasporangium on the scale. So here is the megasporangium. And same thing that you know from before with regard to a sporangium. In the sporangium, you have meiosis occurring. And in this sporangium, meiosis occurs to make spores, the same thing that you've seen before. So we have a spore, and we gave it the term megaspore because it was inside a megasporangium, a megaspore. And this spore is N. Now again, building on what you know before, nothing is new. This spore on the scale, so we'll put our megasporangium again. All of this should be very familiar. The spore is inside the megasporangium. Spores develop into a gametophyte. Same thing that you saw before. So inside the megasporangium, we now have a gametophyte. That is that haploid plant, the other part of the generation. And if you look carefully at the gametophytes of the gymnosperms, we said that at one end of the gametophyte, we have an archegonium. They still retain this from their fern ancestors. And inside the archegonium, you have the female gamete, which is the egg. This whole structure here, the archegonium, the megasporangium, the gametophyte, and what will be, we will say, the incipient seed coat. All of these together, the egg, the megasporangium, the gametophyte, and the incipient seed coat, are called the ovule. So all of those combined together are called the ovule. Important point, because we're going to refer to the ovule again later in today's lecture. And I want you to understand what it represents. Now, following fertilization, that is fertilization by pollen being transferred from the wind, if we carry it to one stage further, and we look at inside the megasporangium. Here's the megasporangium. And here is the seed coat. That great invention of the gymnosperms. What we find in the center of this following fertilization we have the new embryo. The new embryo. This is the new sporophyte, the new 2N individual. Now, there are a couple of tissues that I want to point out to you on this seed. The first is the new embryo, which we said is 2N, but it's surrounded by all this other tissue here. What is all this other tissue? Well, it's megagametophyte tissue. It's tissue which was produced as a result of the spore dividing. This is megagametophyte tissue. And the reason I want you to appreciate that this is megagametophyte tissue is it serves as nutrition for the developing embryo. And for those of you who eat pine nuts or pine seeds, the reason they taste sweet, the nutrition that you get from the pine nut or the pine seed, is mostly megagametophyte tissue. This is what you're eating. This tissue is supplying nutrition to the developing embryo. And you're going to look at this in the lab. You're going to be able to see the developing embryo. So the important point for you to make, to understand, is that within the seed, we have now 3 different types, and this is an important point, 3 different types of genetic material. We have the new embryo. It gets 1 N from the dad, 1 N from the mom. This is an important point. We have the megagametophyte tissue. It's only N. All its genetic material comes from the mom. And then we have the seed coat. It's 2N, and both of the N's come from the mom. So here we have 3 different types of genetic material which make up a typical seed in the gymnosperms, and different portions of the genetic material come from either the dad or the mom, an important point. Now, gymnosperms were a very effective, they were a very successful group of organisms, and that is because they had the seed and they had wind pollination, and they were able to spread to many areas on the earth, and they comprise maybe upwards of 15,000 different species of plants. Sometime between 150 and 190 million years ago, the next step in the evolution of plants took place. And it's a real mystery how this step occurred. And what we can say is that between 150 and 190 million years ago, a now extinct gymnosperm ancestor gave rise to a new group of plants. And that new group of plants is called the Angiosperms. And they are the predominant flora, the predominant plants on the earth today. Now, just like the word gymnosperm, which meant naked seed, the term angiosperm also implies something about the seed. The term angiosperm means seed in a container. Seed in a container. So let's take you back now to the demonstration which we had at the last lecture. Here is a gymnosperm cone, and to just remind you that these seeds are naked, once the scales open, the seeds are not surrounded by any other tissue. If I turn the cone upside down, the seeds just fall out. they're directly exposed to the external environment. This is contrasted with the angiosperms, because what the angiosperms have done is to develop extra protection, which right now we're calling a container. And let's show you that extra protection. Here is an apple that most of you are familiar with. And if we cut the apple down the center, and we're lucky, what you can see is that in the center of the apple are the seeds. These seeds are not exposed directly to the outside. They are in this container. They have this extra protective tissue surrounding them, which is the great advance, one of the great advances of the angiosperms in offering additional protection to the developing embryo, whereas in the gymnosperm, the embryo was potentially exposed directly to the outside. So the seed in the container is this material on the outside, which you know of as a fruit, and this container or fruit wall represents the additional protection that is offered by the angiosperms. So one of the secrets for the success of the angiosperms, and there are about 240,000 species of angiosperms in the world, one of the great reasons for their success is because they have this extra protection surrounding the seeds. The other reason for their success is something that you all know of. And this other reason for their success is that the angiosperms not only developed extra protection, which you know of as the fruit, but they developed at least one other obvious feature. That is, they developed flowers. So with these two remarkable evolutionary advances, the development of this container or the fruit and the development of flowers, angiosperms were able to cover the entire planet, to really confine the gymnosperms to areas where the angiosperms basically could not grow. And what I want to say at the outset for this discussion is that these two are linked. The extra protection is linked to the development of flowers, or to the origin of flowers. And let's now spend some time talking about flowers, because they represent really the place where we can understand where the extra protection comes from. And unfortunately for us, although they looked good when we first got them, I have here a couple of very sad-looking flowers, which should have been upright like so, but for whatever reason were wilted. Now, let's first of all define what a flower is, and then we're going to look at the flower a little more closely. What we can say is that the flower is a determinate, and I'll define that axis. That is an axis with defined period of growth, along which are arranged the organs which make up the flower. So let's spend some time now looking at this axis, which has a defined period or defined duration of growth. So here is the axis. And if you look at a typical flowering axis, as we have here, beginning at the lower end of the flower, we often, but not always, have green, almost leaf-like appendages or organs. So at the lower portion of the flower, we're going to draw this first group of green organs. This is sometimes called, these appendages on the flower are sometimes called a whorl, because they are arranged kind of in a circle around. They're called a whorl. If we identify these appendages, which, as I said, are usually green, these are the sepals. They are the outermost portion of the flower. Moving up on the flowering axis toward the tip, we now encounter the part which most of you know, the most colorful part of the flower. These are arranged in a whorl toward the tip above the sepals, and these are the petals. And they're often, but not always, highly colored. And they represent for many of you, as for example on this rose, sort of the part that attracts you to a flower. These are both sterile appendages. As we move up the axis toward its tip, we encounter another whorl. This whorl is the stamen, and this is where the male gametophyte is produced. The male gametophyte is produced. And finally, when we get to the top of the axis, we encounter the last whorl, which is made up of the carpels. And this is where the female gametophyte is produced. So like the ferns and the gymnosperms, in the angiosperms, which are most of the plants that you see outside, the dominant generation, free living, that means they can make their own food, generation, is the sporophyte. So when you look outside, you're looking at the sporophyte, that is the 2N generation. Now what I'd like to do is to spend some time focusing on the stamens and the carpels, because this is the business end. This is where the reproductive life cycle of the plant really rests. And what I will do is we'll first talk about the stamens, and I'll draw for you a typical stamen, although, as you probably can guess, there's lots of variations on it. So first, let's talk about the stamen. Stamens generally consist of two parts, which are modified, different sizes. And those two parts are a part which is somewhat elongated, which is known as the anther, and a very thin filament, which can be of different lengths, that connects the anther to the flower, joins the anther to the flower. Inside the anther, microspores are made. That is, if spores are made inside of this structure, the anther is a microsporangium. Just building on what you had before. Nothing is new. So the anther is really a sporangium, and inside of that, microspores are made. These microspores are N, so meiosis occurs here. Inside the anther. As with the gymnosperms, the microspores become the pollen. And I'm going to draw a typical pollen grain, and then we're going to return to it in a moment. A typical pollen grain, once it is mature, and this can vary a lot, will have two cells, although not always. And this is just for our discussion here, I'm generalizing. Two cells, one of which has one nucleus, and one of which has two nuclei in them. These two nuclei are called the sperm nuclei. You'll see why in a moment. And this is also, you'll see why, this one is called the tube nucleus. They are all N. So now we've produced the pollen, and it's ready to be distributed and for fertilization to occur. Let's talk about where the pollen goes by looking at the carpel. That is the site where the female gametophyte is made. Typical carpel, again, I'm going to generalize, consists of 3 parts, which are often of different lengths and different shapes, but we're going to make a general one for you. Consists of 3 parts. A flat, or sometimes in swollen top, which then has underneath it a filament-like structure, which then connects to a swollen base. Now, these 3 different parts are given a name. This is the stigma. This is where the pollen lands. A filament-like structure which connects the stigma to this swollen base, which is called the style. And then at the very base of the carpel, the swollen-like structure, which is called an ovary. These 3 together, the stigma, the style, and the ovary, comprise the carpel. So if you were to look at this flowering axis, and you pulled off all of the petals and all of the stamens, and you looked at the very, very top of the axis, you'd find the carpels, and those carpels are made up of these 3 different parts. Now, let's look inside the carpel. If we look inside the carpel, what we can find, again, it depends on the species. For our purposes, we're going to make 4 of these. We find the ovules. Remember, when we talked about the gymnosperms, we said that the ovules contain the female gametophyte, which includes the egg. That is the female gamete. Now let's look a little more closely at the female gametophyte of the angiosperms. I'm going to take you back on a path that you already know, but we need to take that path back. If this is a gametophyte, the earlier step in the gametophyte production, so let's just put here gametophyte. The previous step, which led to that gametophyte, is a spore germinating. Spore germinating to make that gametophyte. What does the gametophyte look like? How many times does this spore have to divide? How many mitoses do you have to have in order to produce this gametophyte? In something like the bryophytes, in something like the ferns, where the gametophyte was pretty large, you probably had to have millions of mitoses in order to develop the gametophyte from the spore. How many mitoses do you have to develop the gametophyte in the angiosperm? So let me draw the mature gametophyte in the angiosperm. The mature gametophyte if we look carefully, we see that it is comprised of a number of cells, just like the fern gametophyte, but a much smaller number. At one end, we have 3 cells located. Each of them has a nucleus. At another end, we have also 3 cells, but one of these 3 cells is highly enlarged. Each of them have a nucleus. And then in the very center, we have this very big cell in the very center, which contains 2 nuclei. All the nuclei are N. They're all haploid because they all come from the spore. Now, of these nuclei, all 8 nuclei and 7 cells, in total, these 8 nuclei and 7 cells make up the entire little plant. The gametophyte now has been reduced from something giant, for example, in the algae, or the liverworts, down to this tiny structure, this little teeny plant, which has only 7 cells in it. This is the gametophyte. The second point that you want to keep in mind is that this gametophyte is not free living. It spends its entire life as part of this flower, never is free living, so it gets all of its nutrition from the sporophyte, from the mom that it is growing on. It cannot undergo any photosynthesis. Now let's look a little more carefully at the cells which make up the gametophyte. There are really only 2 that we have to focus on. The rest of them are not important for the discussion today. The first end cell that I want you to focus on is this large one. This is the female gamete, the egg. It is haploid, just as all gametes would be. The other cell I want you to focus on is this giant cell in the middle. This giant cell in the middle has 2 end nuclei in them. Now, let's talk about fertilization. Let's assume that the pollen has been transferred by the wind or some other mechanism, and we'll talk about those other mechanisms in a moment. As you can imagine, there's lots of pollen flying around outside. How is the plant going to know whether or not it's the right species of pollen which is flying around where it could land on the stigma? The pollen has on its surface recognition signals. These are proteins and other compounds. These are the compounds that make many of you have hay fever. Many of you have allergies because of these factors on the outside of the pollen grains that are necessary for recognition by the stigma. If appropriate recognition occurs when the pollen grain lands on the stigma, and lots of pollen grains which are not related land and they die, if appropriate recognition occurs, the pollen grain germinates. Pollen grain germinates. And if we look carefully at this germination, we find the following. So we're going to draw the carpel again. I'm going to make it a little bit bigger for our purposes. And I'm going to only put, in this case, 1 ovule. I'm just going to put 1 ovule in this carpel. And this 1 ovule contains the little teeny plant. And I'm going to only put the 2 cells that we wanted to focus on before. These 2 nuclei in the center, which are sometimes called polar nuclei. It isn't as important, you know that. And this is the egg. So the pollen has been flying around, and it lands on the stigma, and now the plant and the pollen grain recognize themselves as being compatible. It's the right species. The pollen grain then germinates, and it gets its food and its water from the mom. So here's the pollen grain, and now it germinates. And what does germination mean? Germination means that it starts to produce a long cell, which is known as the pollen tube. Pollen tube. And it grows down through the stigma, and it goes down to the end of the ovule, where the egg is located. Now recall, the pollen grain, we said, has 2 sperm nuclei. 2 sperm nuclei in a typical pollen grain. What do they do? 1 sperm nucleus fuses with the egg. And 1 fuses with the polar nuclei. So what do we get from this particular fertilization procedure? If we look at the products of the fertilization, we have two. First of all, you have the new 2N embryo. The new 2N embryo. That's when one of the sperm nuclei unite with the egg. The other product of the fertilization is you have a tissue which consists of 3 times N nuclei. You have the N nucleus of the sperm and the 2 N nuclei of the pole nuclei. To make up a cell which will develop into a triploid tissue, which we're going to talk more about later on. A triploid tissue, which is 3N. This whole process of fertilization in the angiosperms is known as double fertilization. It is unique to the angiosperms. It is a new process which developed in the angiosperms. How it developed, why there were 2 fertilizations is not understood. There are lots of theories about why this double fertilization process arose, but we really don't know for sure. Now what I'd like to do is to show you a few slides of these various events which are associated with the flower, and then we will move on to another aspect of the angiosperms. Okay, this first slide, if we get the lights down, looks good. This first slide is going to show you a picture of the typical parts of lighting off. No, that's not it. See if that does anything for us. Well, this first slide is going to show you a typical flower, and what I want to point out to you are the parts of the flower. At the very outside, we have the sepals. Most of you are familiar with these, and the sepals are covering up the petals, which are the highly colored part. And then inside, we have the stamens, or the anthers, and they are sort of, they don't last very long once they produce their pollen. And finally, in the very center, we have these mature carpels. I don't know if we can get the lights down a little bit. I don't know if you can get the lights down. Sorry, there you go, perfect. Okay. Now, if we look at the next slide, here we can see now the development, and this is the key, the development of the fruit. For the angiosperms, and this is an important point to put in your notes, the angiosperms, the fruit, that is the structure which protects the developing seeds, comes from the ovary wall. The ovary wall then represents the new innovation developed by the angiosperms to protect the developing seeds so that they are no longer naked. And the ovary wall is essentially the fruit of the flower, a fruit of the plant, and it is the protection for the seeds that are held within the flower. And so you can see as the ovary enlarges, the seeds now become more and more separated and protected from the outside, just like we have in the apple. So this is a really, really important innovation on the part of the angiosperms. Flowers come in dramatic, different sizes and shapes and colors. Many of these you're going to see at the garden. Here is a flower which this is not taken at our botanical garden, but we have this flowering of the garden periodically, and it's usually announced on the web. And it's worthwhile taking a trip up. All of you can get into the garden for free just to see this flower. It's quite dramatic. Here now we can look at the parts of the flower. This is a lily flower, and here are the sepals, 3 sepals alternating with 3 petals. And what I want you to notice is that in this case the sepals are colored, just like the petals. Here you can see the anthers. There's the filament, and here's the anther making up the stamen, and the yellow represents pollen. Now, if we make a cross-section through the anther, which we said is a sporangium, we find that in the lily we have 4 different sporangia, inside each of which microspores are made by meiosis. To make the pollen grains, this is a high power. Here's the sporangium wall on the outside. The whole thing is the anther, and here are the pollen grains being made. And they're made by meiosis and mitosis to make a collection of 4 haploid cells, which are then released. And what I want to point out to you is that pollen grains have very distinctive outer walls. They're very resistant to breakdown, to decay in the environment, and hence they are often used as an indication of finding out what plants were growing in an area many hundreds, if not thousands of years ago, because the pollen wall still persists. And for people who are interested in oil prospecting or geosciences, pollen grains often indicate what plants were growing in an area and whether that's a likely area to prospect for plants. It's also used by anthropologists, for archaeologists, to try and understand what foods and what crops people cultivated. Now, when the pollen grain germinates, we produce this structure, and this is what you're going to do in the lab, which is called a pollen tube. This normally grows down through the center of the style and then reaches the carpel, reaches the ovary. And here you can see the pollen tube with the two sperm nuclei. They actually sort of crawl down the pollen tube, and they then affect fertilization. So this represents now the angiosperms, which are a particularly innovative group of plants, and they're innovative because, we'll go ahead and put the lights on now for a second. I'm going to leave this screen down because we're going to actually show you some more slides in just a moment. So the angiosperms, let's just review this, are successful at least for the first two reasons we gave you. And those two reasons are, number one, they protected the developing seeds in a new way. That is, they made the ovary, which develops into the fruit. And the second big advance of the angiosperms, and we're going to dwell on this advance in a moment, is that the angiosperms made the flower. Now, you might imagine, and I think you would be correct, if you were going to make this flower, even if in this particular case over here, this flower represents a tremendous expenditure of energy, genetic material, in order to develop this particular structure. To what advantage is it for the plant to put all of its energy, or a lot of energy and resources, into making a flower? Why does it just not turn out to be a gymnosperm and not make flowers? What is the advantage of the flower? And basically, what you can say is that the flower is now a mechanism for escaping the need for wind to transfer the pollen. Essentially, what the angiosperms have done now is to develop an organ, in this particular case the flower, which not only will form the ovary to protect the developing seeds, but also is part of a mechanism to allow the plant to use some other way of pollination or fertilization that doesn't involve the wind. And that other mechanism involves insects or animals or maybe even fish, other kinds of organisms. And what this is now is facilitated pollination. The flower essentially is the means for attracting a pollinator. You're going to talk about this more later on in the class, but I want to introduce this idea to you now. Very often, what we find is that the insect or the pollinator and the flower evolve together, each benefiting each other. The flower benefits by having an attraction to the insect so that the insect carries the pollen. And the insect, as we'll talk about in a moment, receives some kind of a reward from visiting the flower and transferring the pollen. So let's talk a little bit about the flower and talk about how it has helped the plants. You recall when we talked about the gymnosperms, we said you needed, for each effective pollination in the gymnosperms, you needed 1 million pollen grains because the wind was random and the chances of a pollen grain landing on a pine cone were very remote. When we use the flower, when we have the flower, which then uses insects or birds to facilitate pollination, we can reduce the number of pollen grains down and still have effective fertilization. So compared to the 1 million pollen grains that you need in the gymnosperms, in the angiosperms, it's estimated you need only about 6,000 pollen grains for each fertilization. So having this facilitated pollination allows for a reduction in the amount of pollen that you have to produce, the amount of resources you have to put into making the pollen, and instead those resources can be made or used in making the flower. So this evolving of the insect with the, in this case, the flower, together to benefit both of them is known as coevolution. Coevolution. And we're going to be talking about coevolution later in the class, and I just want to introduce it to you. One of the prime examples of coevolution is the flower, and I want to give you some examples of this coevolution. It's a fascinating topic, and what I would like you to do, you're welcome to take notes on this, but I'd like to suggest you put your pens down and listen to some of these wonderful coevolution examples. So not only do we produce the flower, not only do we have the extra protection for the seed, but we now have facilitated pollination. And I'd like to tell you a few of these terrifically complex, amazing coevolution mechanisms. The first one I want to mention to you, and I'm going to show you some pictures of it in a moment, is in figs. Most of you have eaten a fig. If you look at a fig and you cut it open on the inside before you eat it, what you will find is that a typical fig is lined with tiny, tiny flowers. So this whole structure is not really the flower. In fact, inside are the flowers, inside the fig. And these flowers come in 3 different types, or actually 2 different types. In one type, the stigma and style and the carpel are long. So we have a long style. And so here is the stigma, and here is the style. And in the other type of flower which lines, the style is quite short. So here is the stigma, and here is the style. Now let me tell you about how pollination works in these figs. A female wasp, very tiny, and if you look at the end of the fig, and I'll show you a picture of this in a moment, the end of the fig is covered with scales that are overlapping. A tiny female wasp, probably the size of a fruit fly, enters the fig by pushing her way through the scales. So she enters somewhere up here at the top. She loses her wings on the outside, and she is carrying pollen with her from another plant. And we'll talk about how she gets that pollen in a moment. It's totally dark inside this, and as she is traveling around inside, she is depositing pollen on the stigmas of the long style and short style flowers. The plant really benefits. How does the wasp benefit? She has an ovipositor, which is a portion that allows her to lay eggs. And she inserts the ovipositor into the flower to lay her egg, with the notion being that as the egg develops, it will be able to feed on the developing seed. But the ovipositor is only long enough to reach down in the short style flowers. In the long style flowers, the ovipositor only goes part way down. So when the egg is here, it dies because it doesn't have any food. When the egg is over here, it lives because it has the food as a result of pollination. These are pollinated, and they produce seeds, but the wasp dies. These are pollinated, and the seeds are consumed, and the wasp lives. So the wasps then mature inside the fig. The males and the females of the particular species mate, and then the males die inside the flowers, never really ever seeing daylight. And the females now are covered with pollen because the little flowers now make more pollen. And the females then leave the fig fruit and fly off and complete the life cycle, being covered with pollen again. How could something like this coevolution type of mechanism, have evolved? For those of us who are vegetarians, you have to give in to the notion that when you're eating figs, you're probably eating some blind male wasps as well. So I don't know whether that appeals to you. Let me show you a picture of the fig and then a couple of other coevolution mechanisms. They're really fascinating. So could you dim the lights again? I'm sorry. If we look at this particular slide here you can see the figs which are growing on the tree, and if you look at the end of the fig, you can see where these scales are. This is where the female enters. Here is the female wasp with her ovipositor. here are the flowers which line the inside of the fig with the short styles and the long styles. You can kind of see that better in this next slide here, where we have flowers with long styles and flowers with short styles, and the egg can grow in one of those flowers and not in the other. The female then fertilizes by inserting her ovipositor into the short and long style, but only in the short style one do the eggs live. Now, perhaps the most unusual coevolution mechanism is found in the orchids. Orchids, as you know, come in a wide variety of colors and shapes and flowers. They are among the largest group of flowering plants. And when you look at a typical orchid flower, it is designed not for us, but it is designed to attract the insect. And we'll talk to you how it attracts the insect in a moment. But let me just tell you what the orchid does. The insect arrives, and I'll tell you why it arrives, and it's a he. And it enters, I'm going to show you a view looking here, it enters into the flower by passing through this passage. It can only squeeze in. It can't squeeze out. And the only way that it can get out is by moving up through the flower. And as it exits the flower, the orchid puts onto its back a small number of packages or containers of pollen. And these containers remain very tightly attached to the bee until it visits another orchid, and then there's a mechanism on the other orchid flower for removing this mechanism. Now, you might ask yourself, what does the bee get out of this? Why should it visit the flower? Well, presumably it could get some pollen, but let me tell you another story of what the bee we think gets out of it. Here are some orchid flowers. Hard to believe these are flowers. They look just like the bees. Here are the antennae. Here are the wings of the bee. Here's another one, which looks like an insect. Or take a look at this one over here. Looks like an insect. What the orchid has done is to mimic the female of the insect and thereby attract the male to come to the flower thinking that it's a female. It probably also makes a scent, which is similar to what the female would make. The flower goes so greatly in its attraction that if you look at the back of the female of the species, she has hairs on her back, and the orchid petals have also put hairs, so that when the insect meets the flower, it's meeting the female. And what the insect is doing is it is mating with the flower. This process is called pseudocopulation. Now, in this process, the flower gets pollinated because the pollen is transferred. And we presume, although we don't know for sure, that the male bee goes away a little happier. So that's the real advantage. Here's another process of pseudocopulation. This is between the grasshopper and an orchid flower. It doesn't look very pretty to me, but beauty is in the eye of the beholder in this particular case. Here's an example of, for those of you who come from the desert southwest, where the yuccas flower at night, they have white flowers, and they make a scent at night if you go out at night. And these flowers are attractive to moths, which are around in the evening. And what the moth does is it lays its eggs down between where the three stigmas of the yucca flower meet, and then packs pollen on top of the eggs, both fertilizing the flower and then providing food for the insect when they hatch as larvae. The last few slides show you, for those of you who come from New England, and this is would be covered now in snow in New England, this is a skunk cabbage. Skunk cabbage is a plant which attracts flies to pollinate it. And the way it attracts flies is that it heats up slightly from the ambient surrounding it, and it volatilizes a compound which the flies find very attractive. And in heating up to release this compound, it also melts to snow, which is directly near the plant. Finally, I just want to show you a couple of more where the flowers heat up and they give a smell of carrion, rotting flesh. For those of you who have been to Australia, and I have a plant up here which is of this type. This is a plant which is like a bottle brush plant, which is pollinated by insects, by animals, small marsupials which crawl over the flower, and when they crawl over the flower to get nectar, they transfer the pollen. And I have one of those flowers up here in front. If you come up and feel it, it has quite a bottle brush feel to it. And finally, this is the question I want to leave you with. This would be like the Pilobolus question I left you with, is that this animal or insect facilitated pollination is very effective. As I said, you can reduce the number of pollen grains down. And yet, even though it is so effective, some of the angiosperms have given up this insect or bird-mediated pollination and have gone back to wind pollination, like the gymnosperms have. And this is an example of the grass, which has reverted in its pollination to wind. And the question I want to leave you with is, if wind pollination is so wasteful with regard to the amount of pollen you have to produce, why is it that a plant would revert and go back to wind pollination and not keep insect or some kind of animal-mediated pollination? Think about that question, and we'll discuss it more later in class. Thank you.